So what I want to do is I want to tie back some of the stuff we've been talking about this week to the work that we learned last week. So you'll recall that this is the receptor tyrosine. Let me get that. Here's our RTK, receptor tyrosine kinase. When it binds something, typically a growth factor, remember that it makes a dimer, and then it autophosphorylates these tyrosine residues, which activate a number of proteins here that I told you I need you to remember, which ultimately leads to the activation of a small G protein called RAS. And again, this is not like the G proteins that we saw in the other signaling pathways. This is made of one subunit, but it's activated in the same way. It exchanges GDP for GTP. And then RAS, once it's activated, will in turn stimulate the activation of what we call the MAP kinase cascade. So the MAP kinase is phosphorylated by RAS, which then in turn phosphorylates the MAP kinase, which in turn phosphorylates MAP kinase, which in turn goes into the nucleus. And I mentioned that there was some activation of these small proteins that are in the nucleus, and those are transcription factors. So these are transcription factors. And transcription factors regulate the activity of a gene by either turning them on or off. Transcription factors. And so guess what these ones do? I told you that it was sort of the leucine zipper. It forms a leucine zipper with Phos and Jun that'll turn on some genes. And guess what it does? You ready? It turns on the cyclin. Did you hear that? So this activated Jun and Phos is going to turn on cyclin D, which you should know by now is going to do what? It is going to bind to CDK4 that will then phosphorylate RB. And now RB, which was preventing the cell from going into S phase, preventing the DNA replication from happening. Now RB is going to take the foot off the brake, and now it's going to be allowed to, DNA replication will happen. So coming full circle, these receptor tyrosine kinases, typically they bind ligands that are called growth factors. And it makes sense. If a cell is going to divide and grow, you're going to have a growth factor. And the growth factors are often released into the bloodstream, and then they bind to specific receptor tyrosine kinases found throughout the body. There's one, for example, for your skin cells. It's called EGF, epidermal growth factor. And then the RTK that it binds to is the EGFR. It's like an alphabet soup. But I thought it was important to sort of link together this idea of what we were studying in terms of second messenger pathways, connecting it to this cell cycle and when the cell cycle gets regulated. I'm going to stop here for a second. I'm going to get ready for the last little bit. So what I'd like to do to finish off this chapter is to bring up together the regulation of the cell cycle with diseases such as cancer. So cancer, by definition, is basically unregulated cell growth or cell division. Things like skin cancer or melanoma, typically it's not the skin cancer itself that causes the issue. Having a tumor form by your skin doesn't really affect much. Skin cancer becomes problematic when some cells break off from the primary tumor site and then invade vital organs. So let's take a look at how that might happen. So you now have the idea of how the cell cycle gets regulated, and it's really tightly regulated in the sense that it doesn't move into that S phase from G1 until everything has been checked. So let's talk a little bit about our RTK. This is our growth factor receptor. Let's use that epidermal growth factor from just a few seconds ago. So typically what happens is it binds the ligand and forms this dimer, and then that initiates the autophosphorylation and then the cascade that results in the activation of phos in June. But let's say that there's a mutation in the receptor such that in the absence of the receptor binding, this thing gets phosphorylated. So there's some mutation that causes phosphorylation events to happen inside the cell in the absence of any sort of signal that's supposed to tell it to go. And this is the case in several types of cancer. So what this is going to do is it's going to initiate the entire signaling pathway to continue to go even in the absence of a signal. And as you might likely think, that's going to lead to unregulated cell growth. Because if we think that this is ultimately good, oh, where's my, I want white. Let's just skip all the little steps about the RAS and MAP kinase, and let's just get to the activation of June and phos. June and phos are going to be continuously active or on because this pathway is on in the absence of a signal. And that's going to then do what? It's going to tell RB, that protein that inhibits the DNA replication, it's going to have, that's going to be oh, I shouldn't do that. Let me just do this. So it will tell RB to allow DNA replication to happen. And that'll happen despite the fact that the genome has not been inspected for any mutations, despite the fact that it doesn't know if it has enough resources, etc. And then this cell will just continue in the cell cycle, right? So we've got, this is not the best drawing, but let's see, here's our G1, our S, G2, M, and that's the division of the cytoplasm. So here, there's no real regulation at the checkpoint, and that cell is just going to continue to divide and divide and divide in unregulated fashion. Does that make sense? Kind of? So one of these proteins, like RB, actually let me pause right here for a sec. So RB, which stands for retinoblastoma, it belongs to a class of belongs to a family of proteins that are called tumor suppressor proteins. And it was identified first because it was the protein that is mutated in eye cancer retinoblastoma. So typically we have a functioning copy of the retinoblastoma protein, and it works to keep the DNA from not replicating until things in the G1 are checked off to make sure that they are prepared to do DNA replication. But in a cancer, if that RB is not doing its job, then it's going to suppress instead of suppressing cell growth, it's going to then allow cell growth to happen and therefore lead to a tumor. So again, normally functioning RB protein inhibits DNA replication until it gets an all clear. If RB is not working properly, if you've got a mutant form of it, it's going to allow the cell to replicate the DNA and pass into S phase. And typically what happens in cancers is this initial mutation is then going to allow more and more mutations to pile up. Because as that next daughter cell comes out of the mitotic phase and enters G1, it's going to have a mutant RB, and any other mutations that might accumulate during that life cycle of the cell are then going to allow it to pass right into that S phase and be replicated alongside with the mutant RB. So it becomes sort of a snowball effect. And one mutation can lead to another mutation, which can lead to another. I'll have one more video lecture on model organisms for studying cell cycle control and regulation. Until then, see ya.